Our next speaker is Kevin Esvelt, professor at MIT Media Lab. Kevin is known for his seminal work on phage-assisted continuous evolution, known as PACE, as well as his work on gene drives for ecological engineering, which he will be discussing today.
Thank you for the kind invitation and to the organizers for putting everything together. So let's talk something that is potentially even more controversial than GMOs in foods. And let's also see if we can get a question to get even more people to raise their hands. How many of you have engineered an organism? Please raise your hand. How many of you have worried that were your organism to escape, that your engineered allele would outcompete the wild type version? Wow, okay, this is actually highly encouraging. Normally, we don't worry about that, of course, because when we're engineering an organism, we're doing it for our benefit, not that of the organism. And in general, we're diverting vital resources away from survival and reproduction to doing whatever we want. And that's especially true in the ancestral habitat. It's one thing to put in herbicide resistance, and then that plant will have a selective advantage on a farm where we're spraying herbicide. It's another thing entirely to have it spread in the wild. But of course, now with modern genome editing, we can in fact do this, or at least genome engineering. How many of you have delivered into a cell a single piece of DNA encoding both the CRISPR nuclease and guide RNAs that target the genome? Oh, that's not so encouraging. All right, hopefully you're doing something like viral defense. But even there, I hope you're not doing it targeting the genome, because of course, if you do this, deliver in both your nuclease and your guide RNA, along with whatever change you want to spread, and your guide RNAs target the genome, then of course, genome insertion will happen per normal for CRISPR. But once you get this in, then you have a functional copy of CRISPR in the genome. It can then cut and edit the other copy. That is, this is one way of getting a homozygote in a single step. But that is not, hopefully, why you would do this. And hopefully you wouldn't do this for any other reason. You would do it because of what happens when this organism mates with a wild-type organism. That is, all the offspring are guaranteed to inherit at least one copy from the homozygous parent. And in the offspring, genome editing happens again. You convert heterozygotes to homozygotes. So when they mate, all of their offspring will inherit your desired trait and the CRISPR system, and on, down through the generations, until in principle, if you designed it correctly, you could edit every organism of the target species. This is not wise. And the real reason is, of course, as we all know, when we're engineering any biological system, it's very complex. We don't understand what all the parts do. And things go wrong. It just happens. Experiments fail, especially in biology. And that means that, in general, we should always be trying to make the smallest possible change that can solve the problem we're interested in. And we should start small and scale up if warranted. So in the case of environmental changes, agriculture and the like, this is why we run field trials. Make the change in the local environment and see what happens. But this is self-scaling. How self-scaling is it? Well, if you run a simulation of a weak CRISPR gene drive system, so this is actually the least effective CRISPR gene drive system described in the literature to date. This is in fruit flies. It has a copying rate, that is, it goes through homology-directed repair rather than non-homologous end joining only half the time. And the rest of the time, it makes a drive-resistant allele that blocks its spread. Middling low fitness cost of 10%, but no cost of resistance. So these drive-resistant alleles are just going to pile up and block its spread. There, simulations show that it still goes up to more than 30% of the local population before it gets outcompeted by those drive-resistant alleles. 30% of the local population is more than enough that any level of gene flow with neighboring populations means your drive system is going to spread into that neighboring population. And from there, it's going to go up to 30% and probably spread into the next one, and the next, and the next, and the next. So here is how you could get eventual spread to every population, even though you're almost certainly not having any ecological effect, just because it never becomes abundant enough in any one population. How many organisms are required to do this? Well, if you run these simulations and you assume no bizarre incompatibility effects between your lab strain and the wild strain, the answer is, well, it can happen with one. This is the maximum this is the frequency distribution, the maximum frequency attained, and you can see the sort of the shaded yellow is the probabilities. And by the time you're up here beyond 10 organisms released, it's going to go. Is this a problem ecologically? Probably not. Most changes that we make are pretty unlikely to have any major ecological effect. Again, it depends entirely on the change, total case-by-case basis, but probably that's not really what we're worried about. We're also not super concerned about biosecurity. And the reason for that one is these things spread over generations. They're slow. You can detect them. Just sequence anything, and if you see a single read that has a eukaryotic expression signal and a CRISPR component in the same read, that's unnatural. And you can't build a gene drive system like this without one. So anything that's slow, easily detectable, and of course can be countered just by building another one, which we've shown works, is not much of a weapon. So we're not worried about that. Why are we concerned? Well, we all do genome editing. Some of us work in more controversial fields than others with respect to genome editing, but we should all be very concerned about whether there is truly public support for what we do. And the reality is that this is too invasive to be practical. Drive systems are extremely likely to spread beyond field trial sites, meaning you can't run that field trial. And the more people and nations that would be affected by any proposed intervention, even malaria, the harder it will be to reach agreement. In the time I've been talking, malaria has killed probably 8 children. And even there, how difficult is it going to be for all of the African countries that harbor the responsible mosquitoes to agree to release a gene drive system to block transmission? That's an unprecedented level of international agreement, especially concerning something as controversial as genome editing of wild populations. And of course, a single laboratory accident could put paid to the long-standing dream of ending malaria by disrupting the transmission through the vector. Again, 10 organisms, and the odds are it's going to take off. And you can imagine the headlines. I don't know how many of you have stock in Editas or Intellia or anything like that, but if you do, you really don't want to see a headline that says, "Scientists accidentally convert entire wild species to GMOs. Is CRISPR to blame?" We really don't want that. And notably, if that delayed target malaria's efforts by, say, a decade, then that would result in 4 million children dying that could otherwise have been prevented. So when you're next delivering DNA into an organism, especially if it's a fruit fly, please be careful. That's why, in fact, we've been really emphasizing this. We need to be careful about this technology. From first describing it before we actually showed it that it worked in the lab, calling for regulation, developing in the first examples in yeast, demonstrating that all of the safeguards that we proposed worked, and then gathering a group of other scientists to agree on what the recommendations for laboratory research should be. We're trying to prevent that laboratory accident that would just devastate the field and even associated fields. But if we want to actually do something useful, other than trying to persuade countries to go ahead and get rid of malaria and schistosomiasis, we really need some way of reducing the number of people that have to agree. In other words, we need a way of confining the effects to a small region so we can safely run a field trial and only need approval from one national government at most. So we need a local drive system. How do you do that? Well, one of the confinement mechanisms we proposed said, okay, the problem here is that it has everything it needs to spread, so let's just separate it. That is, if you build a drive system that is just guide RNAs and in a host that is CRISPR positive, it will work, but it will only work in hosts that are CRISPR positive. So if you release these, then you would have a bunch of these. You'd have your B alleles, but they don't drive. And they cause A to drive, so it increases every generation. But it's really just too slow to be useful. If instead, you separate it into 3 elements, a serial chain, we call it daisy chain, then C drives B, B drives A, and now you're starting to get something, because the B to A does result in an increase in the population. Now C to B does, but as B increases over time, A increases even more. So you get a much more powerful amplification effect in the local population. But of course, natural selection is still here. It hasn't gone away. It's dragging down the frequency of all these alleles, which means the net effect is kind of like a multi-stage genetic booster rocket. The more of these daisy drive alleles you add to the chain, then the more powerful it is, the greater the spread you'll get of your A element. And to give you an idea of the comparative power of these, obviously it depends on the fitness cost and homing rate, but here's your 2-element split. Here's your split drive, 3-element, 4-element, 5-element. And of course, if we go out even further to say 7 elements, then 90% homing rate, you have 7 elements. The most relevant plot here is this one. So what is the frequency you need to release in the local population? How many organisms per population size you want to modify in order to get the frequency to essentially fixation? So 20 generations, I mean, depending on the organism, you probably don't want more than that. So if you follow the 20 line out, you see you need to release for a 5 or 6 or 7 element daisy drive, you need to release about 1 in 50. But the best drive systems in mosquitoes to date have found that the homing rate in the germline of the mosquito is actually 98%. And if you do that, now you're out here in territory of 1 organism per 500 that you want to edit. So this is way more efficient. For comparison, the sterile insect techniques that have been used against mosquitoes, primarily in Brazil and on various islands, and not so much in the Florida Keys, they release 5 sterile males per 1 wild mosquito. So we could be, in principle, 3 orders of magnitude more efficient. But there's another way to do this. Instead of chaining them together linearly, you can just have lots of B elements. So if the first one was termed a daisy chain drive, this is a daisy field. You just have a field of guide RNAs, in this case, driving the A allele that has your CRISPR nuclease and whatever your desired change might be, potentially. So daisy field, say you have just 4 guide RNAs scattered everywhere in the genome that in turn drive the CRISPR gene and whatever cargo you want, make it homozygous. When you have 4 copies of these guides in your initial organisms, they mate with wild type. On average, the offspring are going to inherit 2. And then it's going to be down to 1 and then 0. So you can see the drive runs out of steam. But it still does spread to an appreciable extent. So now we're looking at ways of inserting lots of copies of guide RNAs throughout the genome to quickly create a powerful daisy field drive system that still won't spread indefinitely. So daisy chain, again, you just serially link the elements together where each drives the next. Versus daisy field, you simply have lots of copies of your B element, all of which drive A directly. And they have different strengths and weaknesses. But none of this solves the problem of gene flow across political boundaries. So one of our other projects that we're not actually using any kind of drive system on, we're trying to prevent Lyme disease and other tick-borne diseases by immunizing the mice that infect most ticks. So suppose this works. Suppose Cambridge decided that they wanted to immunize all of the mice in Cambridge. But what if Somerville said no? It's not like we can tell the mice, no, don't wander across that boundary line. Mice don't know that it's there. So how can we do this? We need some way for the genes themselves to tell whether or not they're supposed to be there, and for natural selection to act accordingly. So what we need is we need them to call a quorum vote. If there's a lot of engineered genes in an area, then they're probably supposed to be there, and so they should be favored over the wild type. But if there's only a few, then they're probably not supposed to be there, and we want the wild type to outcompete them. So how do you do that? Well, you can create a genetic quorum by working with haploinsufficient genes. So most ribosomal genes, you need 2 copies for the organism to survive. If you identify 2 of them and you swap their positions, then you end up with a heterozygote. It has 2 swapped copies. When it mates with a wild-type organism, 1/4 of offspring inherit 2 swapped copies and are fine. 1/4 inherit 2 wild-type copies and are fine. But the other half inherit 1 of each, and therefore are imbalanced, and so they die. In other words, it's really bad to be a hybrid. And in an environment where it's really bad to be a hybrid, the phenomenon is called underdominance, whichever allele is in the majority is going to win, because it's more likely to mate with something like itself, and then its offspring will not be hybrids, and they will be more fit. The neat thing about this swap is that it's something that we could spread with a daisy drive, either daisy chain or daisy field, if you have a daisy element in the form of guide RNAs here, and here are your haploinsufficient genes, we swap the positions. Oh, that's an interesting effect. See, this is, when this sort of thing happens, it makes you wonder, should we really be engineering biology, especially out there? Like, really? Oh, OK. Well, let's look at these ones down here then. Whoa. Here you have a daisy element, and you have a CRISPR nuclease next to your swapped ribosomal genes. The daisy element catalyzes the exchange. It cuts the wild type brown, replaces it with recoded green and CRISPR nuclease, cuts green, replaces it with recoded brown and CRISPR nuclease. In other words, all the progeny are guaranteed to inherit 2 swapped copies as long as this daisy element is here. But only half of the progeny are going to inherit the daisy element. Once you run out of daisy elements of one form or another, you're left with hybrids. And again, it's bad to be a hybrid. So the net effect is you could release daisy quorum drive organisms into Cambridge, and then we could monitor how far they spread, release more here, more wild types over in Somerville if necessary to push them back. And where the drive would cause it to spread out towards the edges, and when you get close to the city boundary, it runs out of daisy elements, and then it starts making hybrids, and the hybrids are less fit. And so in Cambridge, you'd have selection for the engineered variants. In Somerville, you'd have selection for the wild variants. That is, any engineered mice that wandered across the border would mate with wild types, create hybrids that would be disfavored. Natural selection would wipe out the engineered alleles. It's not perfect. We would love to have an absolute barrier, but it's much better than what we have now, and it might even be enough. Who knows? But most exciting, we have to face the fact that CRISPR is becoming more and more accessible. There are even high schools now that use CRISPR for genome editing experiments. And while building an actual gene drive system is, of course, much, much more difficult than engineering some E. coli or yeast, the limiting step is the transgenesis. There are also many well-funded high school labs that work with fruit flies. I don't know if any of them do transgenesis, but conceivably they could. At the very least, there's thousands and thousands of people that can do it. And that number is probably only going to grow. After all, we can all see the incredible potential of genome editing. So sooner or later, it is highly likely that someone is going to edit the environment without the consent of the affected community. We desperately need a way of restoring that population to wild type. So how do you do it? Well, daisy restoration drive is basically that daisy quorum drive I just described that's engineered to overwrite its target. But what we really need is a way of overwriting and replacing every copy of a rogue invasive CRISPR gene drive, the kind that spreads forever. So how can you do that? Well, if you take a daisy quorum drive and you add some guide RNAs that piggyback on the nuclease of the rogue drive system, and your daisy quorum uses an orthogonal nuclease, then in the presence of the rogue drive nuclease, it will spread like an invasive CRISPR drive system. It will spread indefinitely, and it will spread all of the daisy elements. It will drive even the daisy elements not as well as your actual daisy drive nuclease. So if you parachute in a few mice, assuming it's mice that have been edited here, into the area that is where you've detected the unwanted rogue drive system, then your daisy drive will begin spreading like an invasive drive system until it overwrites every copy of the invasive drive system. But in order to do that, you can't allow the invasive drive system to continue spreading through wild type. You can't just play catch up. It will always have more wild type nearby. But with this one, when it hits the wild type population, it still keeps copying and spreading through the wild type and immunizing them against the rogue drive system. The difference is it relies on its daisy elements to do that. And so it starts losing them. So in other words, it spreads efficiently and overwrites every copy, then spreads a little bit, immunizes the nearby wild type population, so it has a chance of getting every copy of that unwanted drive system. And then you have this daisy quorum effect on the edges in particular. It's bad to be a hybrid. So if we just release a lot of wild types, or better yet, since all of these have CRISPR nucleases, your daisy nuclease, if you drop in a suppression drive element, which is just guide RNAs that targets, say, a recessive female fertility gene, so that any female offspring of 2 parents who are carriers are born infertile, then you can selectively suppress the engineered population. As soon as it's in the minority, then it's at ever more of a disadvantage due to the quorum effect. All the engineered changes are linked to the quorum effect, and so population becomes wild type. Is this Rube Goldberg? Well, yes, it's Rube Goldberg. It's totally Rube Goldberg. But it's Rube Goldberg where every last step is CRISPR genome editing. So any other biological system, I would say there is no way we have a chance of getting this working. But because it's CRISPR, and CRISPR is frankly magic, I think this has a good chance of providing a way to ensure that the shared environmental commons is whatever the people who live there want it to be. That does not allow single actors to unilaterally decide what the environment should be like. And perhaps even more excitingly, this isn't limited to invasive rogue drive systems. This is something that we could use to potentially restore the wild type any population with any engineered genes at all, whether they leaked from some other species that we meant to engineer or whatever other reason. If it's drive amenable, we could potentially restore it to wild type using a method like this. And more broadly, this ability to edit wild populations could do a lot of good in the world. And not just for humans, although there are certainly many health applications, from mosquito-borne diseases, tick-borne diseases, to parasites that are sexually reproducing themselves and could be targeted directly, to even animal reservoirs of disease. What about animal welfare even? I mean, right now there's a lot of kittens that starve and freeze in the wild out there. And we spend a lot of money adopting them, but there's always more. Wouldn't it be lovely if we could ensure that every kitten was born, came into the world destined for a warm and welcoming home? Wouldn't it be lovely if we could, say, remove the invasive mosquitoes and rats that are driving species extinct on islands? And I could also say, wouldn't it be lovely if we didn't need to spray our crops with herbicides and pesticides in general and instead just engineered the pests to suppress the local population? Or better yet, tweak them so they just didn't like the taste. But if we're going to do that, frankly, I think this needs to come last. These need to come first, because priority is tackling problems that have obvious benefits to the typical person on the street. And again, since I began talking, we're probably up to 16 or 20 children that have died of malaria. So problems like that have to come first. But more broadly, this is a technology that can engineer the shared environment. And that means that if you develop this behind closed doors, you're directly denying people a voice in a decision that could affect them. And I think we need to use this as a test case for a new way of doing science. That is, one where we do pre-register our proposals so that everyone knows what the field is at least attempting to do. And that means, because based on the few reactions of a few of you, who are probably not so keen on sharing your best ideas so they can be scooped by some other laboratory who has more money in hands, we need to change the incentives governing science, at least for this field. And there are a number of ways we can do that we're looking into, from the journals to the funders to even intellectual property. I mean, the Broad took a powerful step by denying Monsanto the right to use CRISPR for gene drive explicitly in that licensing contract, as well as for Terminator technology and other things. So IP ethical licensing is a potentially valid tool that we could use to change the incentives governing particular fields of science. And this is the field where we need it most. We've gone ahead and pre-registered all of our experiments involving any form of gene drive on bioRxiv, and more recently on a responsive science site. And we're trying to adopt an approach where we work with communities to decide what exactly they need so that they can help guide the technology from the very beginning. And this is in large part because I don't trust myself. Technology is becoming so powerful, and it's so difficult to really apprehend the possible consequences, especially when you combine it with advances in other fields. I mean, look here, CRISPR-based gene drive. Austin Burt, who is an absolute genius at Imperial College, who's been leading the target malaria effort, first proposed that we could use homing endonucleases like ISKI1 from yeast to edit wild populations using exactly this form of gene drive in 2003. But he never imagined that we would have CRISPR and that we could therefore potentially apply this to many different species so long as they're sexually reproducing. It's hard to apprehend consequences. And so by openly sharing our proposals before experiments begin, we give the rest of the scientific community a chance to not only decide what they want to do in an open and rational manner? Do you want to intelligently collaborate or intelligently compete, as opposed to the blind paranoia that characterizes most of the CRISPR field? Do you want to, we can actively invite concerns from both the rest of the community and also from local communities of citizens who might be affected. We can actually request guidance from the sites that might be candidates for early field trials and ask the people there what they would like to see. Of course, arrange for independent assessment of any of those field trials, because just like the last person you want to test a hypothesis is the person who proposed it, same goes for evaluating a technology. And lastly, we can, above all else, start local and only scale up if warranted. Thanks in particular to all my students and collaborators that have worked with me over the last few years on this, various funders, but especially to my lab, who have really gone out on a limb. They're the real heroes of this story because they are willing to share their best ideas and, at the moment, risk somebody else scooping them. So thank you.
So we can take a few questions.
In this scenario, like the quorum scenario, does this essentially require people continuously monitoring this and adding or taking away something from the system, or will this become a self-sustaining thing?
Great question. You definitely need to monitor in order to know what's there. So just because you can unfailingly detect a drive system present in a given species, I mean, my hope is that advances in sequencing will eventually allow us to simply sequence the river at the bottom of every watershed and detect any drive system present in any species that lives in that geographic region. Worst case scenario, we have to sequence the populations that we're concerned about. And that's not something that is a priority for the next 5 or 10 years, but eventually it's the sort of thing that, well, let's just say that ecologists should rejoice because they're going to get on the Department of Defense gravy train.
Are you concerned that this restoration through a gene drive would eliminate the diversity in the wild type?
Yeah, so should we be concerned about gene drives removing diversity in the wild type? So mostly what the drive system does is it changes the one gene that you're targeting, or some combination of genes. It's possible that if you have an off-target, that's most likely to just lead to a fitness cost. But you can imagine that an off-target might create a mutation somewhere else in the genome and then drive it through the population. But for the most part, you're not affecting the vast majority of the genetics. You're just affecting those that you intend to. So hopefully you wouldn't do it at all unless you're sure that altering at least that allele in that particular local population is a good thing, and ideally through local field trials to test that out first. More broadly, that is one reason, very excellent reason to be concerned about the self-sustaining invasive version that spreads indefinitely, because there you probably are going to affect every organism of that species. Yeah.
So playing devil's advocate here, you could say that in the 1970s, Cambridge was shit scared of the common DNA technology. And so how do you contract those kinds of arguments that this is something that will not really be as bad as it can be?
Right, so how do we, given the moratorium from the 70s on recombinant DNA that Cambridge imposed, how can we ensure that that doesn't happen? Well, I'm
just saying that maybe it's not as big a concern.
So again, we now know that we don't really have to worry about SV40, recombinant SV40 spreading through populations and causing plagues. And we know that recombinant DNA is not going to break evolution. This doesn't break evolution. Gene drives are natural. They occur in just about every species on the planet, including us, because a transposon is just a different form of gene drive system that spreads even though it does not benefit individual organisms or the rest of the genome. So it's not really anything profoundly new and different. I don't think it's particularly threatening at all. It's not a good weapon, and most changes are not going to have ecological effects. Even if they do, they're going to spread slowly, and hopefully we can perfectly reverse them as needed. Now, it's true that with an ecosystem, if you build a dam, and then you knock the dam down, it's not necessarily going to go back to the way it was before. But that's true of anything, right? That's true of anything at all. So I'm not concerned by anything other than the risk to public perception, because we are, to some extent, in a hole when it comes to the public trusting us. And in many areas, frankly, I will give this to, say, Friends of the Earth and ETC group, whom I disagree with on almost everything. But they do have some valid concerns about the way that we do science. I think that by being more open and responsive and actively reaching out to communities, saying, please help keep an eye on us, help think of things that we might miss. You're the ones that live there. You know more about the environment. Figure out what might go wrong, and then we can hopefully address it in the early stages of technology development before it happens. And then there is no substitute for good communication with the local community. In about 3 hours, I'll be addressing the Cambridge Biosafety Forum and training the IBCs for local biotech companies and universities. So doing that kind of local outreach and communication is absolutely essential.
How has the response been to the Oxitec experiments and limiting and all that? Has that been received positively by political groups in other countries?
Oh, so Oxitec? Yeah, so Oxitec makes the sterile male mosquitoes. And those have been received incredibly positively in, say, Brazil, where there is a lot of dengue. And the people say, wait, you can do something about mosquitoes so we don't get dengue? That's fantastic. In areas like the Florida Keys, you had a recent vote as to whether or not the trial should go forward in Key Haven. The citizens of Key Haven said, no, it should not go forward. The citizens of the rest of the county said, yes, it should go forward in Key Haven. So in that case, what they're going to do is they're picking another section of the county that had voted yes to Key Haven, and they're going to release it there without asking them again whether they wanted it. So this is the NIMBY effect. We know that it is real. But at the same time, I don't think they're doing anybody any favors by moving ahead in a community that said, yes, but only over there. I think the Keys have not had a dengue outbreak for 7 years. And even then, it was just a couple of people. They just don't see it as a large enough problem to take this step. And there have been a lot of missteps by the company in reaching out to the community from an early enough stage. I mean, we have this idea as scientists that if we explain to people what we're doing, then of course they'll support it. Because what we're used to is we say, okay, give me all the available evidence, and I will look at it, and then come to a conclusion about what I think is right. But that's an appallingly hubristic attitude that assumes a great deal of expertise in the relevant area. That's just not how normal people think. We're sort of brainwashed into thinking that way. Normal people just want to know, so first of all, who are you proposing this? Why do you want to do it? Who's it going to help? And what's it going to cost? And who's going to pay the bill? That's what they want to know. And that's a much more reasonable set of questions, assuming that somebody else has the relevant expertise, wanting to know that whoever that is has access to all the data. I mean, this emphasis on transparency and openness, it's not that we expect the vast majority of citizens to even go and look and see if it's there. They just want to know that in the event that someone who does have the relevant expertise takes an interest, then they will be able to see everything that, say, the regulators are seeing. And above all else, I cannot stress this enough, we absolutely have to stop relying on the regulators as a proxy for community approval. Because the FDA only invites comments at the just prior to product approval, when it's far too late to actually change anything about it. And then they invite comments from the public. 90% of them are questions of ethics and values. And the FDA throws all those out because they say, well, you know, our mandate is only to evaluate empirical data and likely outcomes. And so it's like they invite people with their opinion, and then they say, nope, these opinions, not relevant. That's just not good community outreach. So we need to do better as the scientists from very early on addressing these kinds of issues, because at the end of the day, we should hold ourselves morally responsible for all the consequences of what we do. And in a world where it's getting ever harder to predict those consequences, that means we need to invite other people for help.
Great, let's give another round of applause.